To introduce her is her mentor, Julia Rose. Julia's been with TNC for 7 years, and her area of expertise is corals, which you're going to learn a little bit about here. And she also is working statewide to come up with a plan to restore our coral reefs when damage happens due to storms. She, as a side note, is also a Hilo graduate. And when she's not saving our coastal reefs, she is sailing and it's basically playing in the water, and also with her dog. All right, so take the floor.
Thank you, Lisa. Thank you for having me, and thank you for that introduction. And I am very honored today to introduce Mikayla Barnwell. And I wanted to share just a little bit of context with you all first to not only talk about Mikayla and her contributions to our team, but how much her contributions have actually meant statewide, and to sort of the partnership network that is forming organically around coral restoration in Hawaii and across the Pacific. So in 2015, after the global mass coral bleaching event, there's been an ever-increasing acknowledgement that we do need to take we need the ability to take active interventions in addition to reducing our local impacts if we want our reefs to be around in the future. So since that time, in 2019, the Nature Conservancy entered into a cooperative agreement with NOAA to do planning throughout the Pacific, because this was kind of a shift in thinking for the Pacific in particular. And so since then, TNC has done planning in Guam, American Samoa, the Commonwealth of the Northern Mariana Islands, and also Hawaii. So I was on the statewide planning team for Hawaii, and in addition to planning, after that, we had committed to do pilot projects of coral restoration. But this was also a shift for our team. We are, team with a really strong science background. We do a lot of monitoring, and what we didn't have on our team was experience in actual coral restoration. So I first met Mikayla in, I think it was November, maybe October of 2021 over Zoom, when Lisa connected us, and she was interested in getting involved with our team, particularly in the South Kohala Coastal Partnership. She wanted to do watershed environmental education. One of the first things she said on that Zoom call was, "Oh yeah, the Nature Conservancy. I worked for the Nature Conservancy in the Caribbean doing coral restoration." And this was right at the time when we were sort of transitioning from, okay, we're doing statewide planning, it's all very theoretical, to now we are going to do some pilot projects and actually get in the water. So my ears perked up instantly, and I stole her. No, what I actually said was, oh, we're getting ready to do some pilot projects. We're building a capacity across the state for coral restoration, and I could really use some help. So Mikayla joined our team at that time as an intern, bringing on more experience in the work that we are planning than any of our team actually had at the time. And so since that time, we started your internship in 2021, and we actually ended up hiring her as a full-time team member last year in August. So since August, through some sorcery that I do not fully understand to this day, she has managed to be a full-time master's student and be a full-time member of our team. And she has substantially contributed to all of our projects. And she is, I'm sure, going to share about all of the work that she's done. Keep in mind, it's even more than she's telling you. We've thrown her into the deep end on all sorts of things, and she has. I've been honestly blown away by her project management, her organization skills, her team building and work ethic. She's a huge asset to our team, and I am really excited to call her not just you know, she's not just an intern, she is my valued coworker, team member, and I look forward to working with her moving forward. So thank you and welcome, Mikayla.
You guys see me over all these Thank you, Julia, for that wonderful introduction. I'm super grateful to be a part of this team and have been brought on as a full-time member and to work with all of you and the support from my cohort members and everyone else in the audience, as well as all my friends and family watching on Zoom. And so like Julia said, I'm going to be sharing about the work that we've been doing on these pilot restoration projects and some of my contributions for those.
Here we go.
Thank you. All right, so aloha, good morning, everyone. My name is Mikayla Barnwell, and today I'm going to be talking about place-based coral restoration in West Hawaii, developing a guide to planning and implementation. So as Julie said, for my professional master's internship, I've been partnering with the Nature Conservancy's Hawai'i Island Marine Team, which is part of their Hawaii and Palmyra chapter. And so today I'm going to be talking about the work that I've been doing with them to help develop 2 pilot place-based coral restoration projects here in West Hawaii. So I'm going to give a bit of a background, some background knowledge, why we're doing this, why it's important, kind of how it came to be. Also kind of our planning process thus far, what we've gone through, some highlights as well as some challenges, and then what some of our next steps look like. So just to give you guys a quick background before we get into the planning process, I wanted to talk a little bit about coral biology and coral reef ecosystems here in Hawaii. So corals are a colonial organism. So they are covered in this living tissue layer, this yellow-brown mat that you see, that's their living tissue layer. And so within that tissue, there's hundreds and thousands of individual tiny polyps, as shown in this drawing right here. And so in the tissue, in their polyps, they house a symbiotic microalgae called zooxanthellae. And the zooxanthellae are primarily responsible as the main energy source for these coral organisms. They photosynthesize, and then that energy gets transferred to the coral as their energy and food source. And so underneath this living tissue layer is a really thick calcium carbonate skeleton that's super structurally important and adds complexity to the coral reef ecosystems. So here in Hawaii, coral reefs are super important. They house a wide diversity of marine life, many of which are endemic to Hawaii, which just means that they're only found here in Hawaiian coral reef ecosystems. Not only are they important as a food source for other marine organisms, but also for the coastal communities that live around coral reefs with their subsistence-based fishing practices. Coral reefs also act as a natural protector, a natural barrier to those coastal communities. That structural complexity from their skeleton helps to dissipate wave energy from big swell events, from different weather systems, helping to protect that community infrastructure along the coast. And then here in Hawaii, coral reefs are also very culturally important. In the Kumalipo, which is the Hawaiian creation chant, corals are described as an original ancestor for different organisms, as well as ourselves. So it's super important that these organisms are valued, protected, and restored. So as important as these ecosystems are, global climate change is negatively impacting these reefs. Increased water temperatures is leading to more frequent and more severe coral bleaching events, and coral bleaching causes the coral to lose that symbiotic relationship with the microalgae. The zooxanthellae leave the tissues and leaves them a bright white color, and also leaves them without their main source of energy and food, and so it often ends up in mortality for these organisms. And so within the last decade, there's been 2 pretty major bleaching events, one in 2015 and one in 2019. And so this example that I'm showing you here is an example from before and after the 2015 bleaching event. These events led to significant decline in coral populations throughout all of Hawaii. And so these photos were taken as part of the Division of Aquatic Resources. So the state has some long-term monitoring sites in Kealakekua Bay, which is in the west side of Hawai'i Island. And so on the left is before that 2015 bleaching event, and on the right is from 2017. And so if you kind of use these coral colonies here as a reference, you can see the difference from before and after that event. So there's a significant loss in coral cover, but there's also some major erosion that's happening because there's no more live tissue, no more live coral helping to support that. And so it's really damages like these that show a need for coral restoration. So coral restoration in general is an avenue of ecological restoration that focuses on restoring degraded reef habitats. And so this can happen in a passive manner, which is more of like establishing marine protected areas or initiating fishing bans, but it can also happen through active methods such as coral gardening, which involves taking coral colonies, fragmenting them, outplanting them back on the reef as if you were planting in your own garden, except it's underwater. And so coral gardening is a pretty common method used globally. It can happen on land in a lab facility, or it can happen underwater in a coral nursery setting. And so this example here is a common coral nursery structure or coral tree used in the Caribbean. And so the methods that I'm going to talk to you about a little bit later are going to be more in water active restoration, but with a different structure that's more tailored to our environment. So as Julia mentioned earlier, kind of with these devastating damages from the bleaching events in 2015 and 2019, it was increasingly obvious that action needed to happen and restoration needed to take place to be able to restore these reefs. And so entities from across the state got together in 2020 to start this planning process of how to address these damages. And so with that, they developed a restoration action plan with a series of goals. One of which being kind of a driving factor behind the pilot projects that we're working on. And so this goal here, to build capacity, to develop, test, and apply restoration methods that enhance the resistance and recovery of coral reefs impacted by bleaching. It's kind of a driving factor behind the work that we're doing. So in that planning process, they also identified some priority geographic areas of where this work should happen based on impacts of coral bleaching, as well as a slew of other factors. And so here on Hawaii Island, the two locations that were identified that we'll be working in are Ka'upalehu and Kealakekua. And so in addition to the effects of coral bleaching, there's also other conservation work happening in these two areas that will benefit the restoration work that we're going to be doing. Ka'upalehu right now is in the middle of a tri-weight fishing ban excuse me. And so it started in 2006. And so for 10 years, there's no fishing allowed in that area. And it was actually an idea brought forth by the community because they noticed a decrease in the fish populations that they rely on. So they're in the middle of that fishing ban. And then Kealakekua is a long-term marine life conservation district that also is a no-take area. So that additional factor on kind of restoring the fish populations is complementary to the work that we're going to end up doing in these areas. So an important factor of this project is using a place-based approach. Not only is this work important for ecological restoration, but also for those communities in the areas that we're going to be working in. The community members in both of those areas have valuable knowledge of place that is super instrumental, including in our planning process and all of our decision-making aspects of this project. And so as we move forward, it's really important that we bring along and highlight those voices in the work that we're going to be doing. So first in our planning process is figuring out how exactly we want to design this restoration project. What do we want it to look like? What types of methods are we going to use? And how are we going to go about that? And so including community from those areas is super important in this step as well. So the first thing that we did in early 2022, we hosted a series of planning workshops. We had a coral hui, and we worked with different entities as well as organizations in those respective areas. So we had representatives from the Nature Conservancy, Hawaii Institute of Marine Biology, NOAA, Division of Aquatic Resources, or DAR. And then in Ka'upalehu, we had members from the Ka'upalehu Marine Life Advisory Committee. And in Kealakekua, we had members of an organization called Kapu Kapu Ohana, which they originally helped to create a community action plan for that area. And so volunteers from these two organizations, as well as members from all of these entities, came together to make decisions on our project. And so kind of the main decisions categories that came out of these workshops were within those priority geographic areas, focusing and finding specific restoration sites at each of those places, identifying what are target coral species that we're going to be working with for this project, and then solidifying what types of restoration methods, what types of outplant methods are best for the locations and what we want to use. And then also kind of identifying some long-term goals, talking with community members as we work towards implementation, move past that, thinking about scaling up, what's important for them to see, like, what direction is this project going to go? What do they want to see come out of this project? And so talking about that with them as well. So as we go through kind of our experimental design that I'm going to share in just a moment, we kind of identified a few different research goals, research questions that I wanted to highlight first, because I think it's important as we go through these design components, recognizing that coral restoration here on Hawaii Island is relatively novel. And so a lot of this project is working with community, working with these different methods, figuring out what works best for our Hawaiian coral species, what works best for our environment here. Restoration that happens here is going to be very different than restoration that happens in the Caribbean or other parts of the world. Our corals grow differently, and so really kind of figuring out what works best for where we are. So we'll be looking, is there a difference in survival between nursery grown fragments and directly outplanted fragments of Parietes lobata? Is there a difference in success between fragmented corals and stabilized corals? And what is the cost benefit ratio between nursery grown fragments, directly outplanted fragments, and stabilized colonies of Parietes lobata? And so thinking about all that we're going to learn through this, I think it's important that we outline the work we're going to do as the beginning of a framework that could be used or modified by other organizations or by community members that are looking to do coral restoration on Hawaii Island. So within our priority geographic areas, so we have our two coral hui groups in these locations. We went through a series of site visits with our community volunteers from those two organizations to figure out where best in these two areas do we want to see restoration happen. So in Ka'ukalehu, we're going to be working in Kahubai Bay, which is right in front of the Four Seasons and Kona Village Resort. And in Kealakekua, we're going to be working directly in Kealakekua Bay. And so those photos I showed you initially of the coral bleaching effects were taken from this location here. So it does show that it's a little bit of a challenging habitat, but also really highlights the need for this work to happen in that area. So we do have two locations, but because of the amount of work for implementing each one, we're taking a staggered approach. So we're going to be focusing on Ka'ukalehu first, and then kind of using what we learned from that to transition into implementing to Kealakekua. So within each of these locations, I wanted to show you a snapshot of kind of how our restoration is going to look along the reef. So we've got a series of different plots, 10 by 10 meter, half of which, 4 of them will be restoration plots where we're doing direct intervention or impact. And then 4 of them will be control plots. And so that allows us to see how the environmental conditions are impacting kind of natural reef areas without any intervention, and comparing that to the success of our restored areas. And so all of our restoration plots will be kind of around our nursery site, which I'll get into in a little bit. And the corals that we're going to be using in this project are only going to be coming from each respective bay. So Kahubai Bay or Kealakekua Bay. We're not going to be taking corals from other parts of the island and bringing them in to use in restoration in these two locations. It was really important for the community members that the corals that we use stay local to that area, but also it's important to minimize the spread of any coral disease or aquatic invasive species. So the species that were decided upon in our coral hui meetings, we have kind of 3 main species across the 2 sites. So Pohakupuna or Parietes lobata, Koa, Passalopora meandrina, and Pohakupuna Parietes compressa. And so we had conversations with our community members in our coral hui workshops to figure out what corals have been impacted most by bleaching, but also what coral species they wanted to see us work with. And so for this initial pilot phase in Ka'upalehu, we're going to be focusing on Parietes lobata and Pasmawa meandrina. And then as we transition to implementing in Kealakekua, we will add in the third Parietes compressa as well. But in addition to kind of these reef building species, it was important for the community to see an increase in coral diversity as well. So maybe some of the more rare coral species that we don't find as often, trying to propagate and repopulate those. So we have a selection of opportunistic species that as we're going along to collect for our primary species, we will work on kind of collecting those as well and opportunistically outplanting and propagating those to help with diversity. So the community also decided that we did want to use a coral nursery setting. And as I mentioned earlier, coral restoration that happens here is going to be very different than coral restoration that happens in the Caribbean, where they can have PVC trees in the water. Those would not last here with the different swell and wave events that we have. So it was important that we design a structure that would be able to withstand kind of those big swell events and protect the corals that are on our tables. We don't want to do any more damage than we're trying to restore. So the design or the goals of our structure here is a robust, heavy nursery table. So we've had these 4 kind of cement corners as the footings of each table, and then a light kind of fiberglass frame as the main portion. So we want really heavy and low surface area, and we have a modular approach with this, and so we'll have nursery like trays that we can take on and off the table as needed if we need to kind of switch out the corals, or if there's a storm event coming in and we need to move the corals to a safer location, we'll be able to do that as well. And so part of this is in using a nursery structure for the different aspects of our project, figuring out, is this the best structure to use as well, and thinking about that as we move through the project. So for getting into the restoration methods for propagating and outplanting, we have kind of 2 main categories that we're going to be working with, stabilization and fragmentation. And I just wanted to quickly say that when I talk about outplanting, I'm essentially talking about taking whole pieces or smaller pieces of coral and gluing them back onto degraded reef areas. Typically, we use a marine epoxy or a cement mixture. So when we outplant them, we're just gluing them back onto the reef, essentially. And so for stabilization, we'll be using our coral fragments that we find. And I forgot to mention earlier, but the corals that we do use are going to be considered corals of opportunity. So we won't be taking any whole coral colonies off of the reef. We'll be looking for ones that have already been dislodged and they're kind of rolling around in the sand, and without intervention will typically lead to mortality. So we'll find those whole coral colonies, corals of opportunity, and we will directly stabilize them into our restoration plots. So finding colonies, outplanting them as they are as whole colonies into the reef. And then for fragmentation, finding those corals of opportunity, but cutting them up into smaller pieces before we then outplant them back onto the reef to increase the biomass there. And so within our fragmentation, we have kind of 2 methods that we're going to be working with, which are dictated by the morphology of the coral. So you can see the example down here. This is a cauliflower coral. It has those kind of like thick, nubby branches. And so based on the way that it grows, using more of a hammer and chisel method, where we can get a bigger fragment and get kind of the tops of the branches that include that essential growth skeleton, so that when they do go through their grow out method, they can continue that branching growth method. And then we will also use a method called microfragmentation that uses this fun specialized aqua saw that I will explain. So our second one is microfragmentation. I promise I'm having fun in this picture. I'm just really focused. And so microfragmentation also involves cutting corals into smaller pieces, but this involves cutting corals into much, much smaller pieces, about between 1 to 3 centimeters usually. And so we'll be focusing on about 2.5 centimeters. And so this is beneficial for those boulder species that have kind of like mats of tissue and maybe irregular shape. We can cut them down into those small pieces with this saw. And then doing so helps to increase their growth rate. And so when you cut corals on different margins, cutting that tissue activates rapid tissue growth. So as we cut it into small different pieces, cutting different margins, we're activating that tissue growth on all sides of the fragment to help kind of get more coral tissue faster. And so from our microfragmented corals, they will either go one of two places, directly outplanted into our restoration plots and these fun little arrays that I'll explain in a second, or they will get staged in our coral nursery area. So we have these little PVC trays that we will attach to our table and glue the fragments onto cement pucks, where they will sit on our nursery table until they're able to grow out and cover the surface area of the cement pucks. And so in doing this, it's helping to look and see how using a nursery table contributes to the success of our corals. If it's needed, what are the benefits? Is it helpful for this species? And as we kind of find opportunistic species, is it better for some than the others? So really kind of figuring out the best way to do this here. And so once the corals on our cement pucks on the table have grown over and cover the surface area, they will get outplanted as well. And so the way that we are outplanting our corals from microfragmentation is in a cluster design. And so this is really based on the average growth rate of our boulder coral species. It's pretty slow. It's like 1 to 2 centimeters a year. And so it takes a really, really long time to get a big reproductive size coral or even a nice adult size coral. And so in doing this, when we outplant them in these different arrays, it's kind of based on initial size. So they'll be smaller if we directly outplant them, and be a little bit bigger if they're coming off of the nursery. But they'll be in a 12 centimeter diameter array. And the goal is to get tissue fusion in 1 year. So by spacing our fragments about 1.5 centimeters apart, that rapid tissue growth over a year will hopefully end in fusion. And then at the end of the year, we'll have a 12 centimeter coral colony, which really accelerates that growth method and allows us to get bigger corals much faster. And so then within 3 to 5 years, we'll hopefully be able to get a reproductive sized coral. So once all of our corals are outplanted into our plots, then we can collect data on them and start monitoring. And our main method for monitoring that we've been using is called structure from motion, or SFM. And so this involves going over our restoration plots that we have and swimming in essentially a lawnmower pattern and a grid, and rapidly taking photos with these nice fancy underwater cameras here that we will then bring back and upload to the computer in a program called Agisoft Metashape. And this program will take all of the pictures from our plots that we've taken and stitch them together into an orthomosaic. So this is a snapshot of an orthomosaic that we made from some preliminary baseline monitoring we did a few months ago. And so using this to help monitor our corals, we can kind of track the success of them over time at different time points, as well as look at survival, measure surface area growth, and see how our different restoration methods are impacting the success and growth of our coral fragments. So now that we figured out what we want to do, how we want to do it, what it should look like, now it comes to preparing to actually implement it, get all of that stuff in the water. And so the next few steps or aspects that I'm going to be talking about, I've been able to be pretty involved in, which was a huge learning curve for me, because as Julia said, my coral restoration background is from programs that have been already established. And so this has been a really cool full circle to kind of go back and help on the project management side and planning aspects of this project. So everybody's favorite part about project is permits, which have been a huge challenge for us at this point. It is, as I said, it's not everyone's favorite part, but I do think it is an important aspect to highlight. It's crucial and required for any coral restoration project or any project that involves handling like live organisms or installing anything. And so understanding the process and the different avenues of this is critical for us, as well as other organisms that are looking to, organizations that are looking to do this type of work. So we've had 4 main permits or consultations that we've been working with. Division of Aquatic Resources, or DAR. We've been applying for a special activities permit. So anything handling the live coral, collecting coral, fragmenting, stabilization, outplanting, all of that is going to be covered under our special activities permit. Our Army Corps of Engineers permit nationwide. So the work that we're conducting in navigable waters, structure deployment. So any of the work that we're doing and installing our nursery table, as well as anything that we need to install for our permanent restoration and control plots would be under this. And really exciting news, we found out on Monday that our Army Corps permit was approved, which is really exciting. And then for the state's Office of Conservation and Coastal Lands, we have a site plan approval. So really any of basic data collection, research activities that's impacting resources or land for more than 30 days is going to be covered under the OCCL permit. And then lastly, a state historic preservation department, or SHPD. We have a consultation. So our project is federally funded by NOAA. And so using those federal funds in the areas that we're working in kind of dictates this consultation. And so once all of these have been approved and we have them in hand, we can move forward in the next step. So the National Environmental Policy Act, NEPA review process also have been a huge learning curve and lengthy process. And waiting for each of these to come in has added onto our timeline of this project. And so it's just important to understand the different steps involved in this. Another kind of similar lengthy process to go through when using this for our nursery tables, because it is so robust, we have to contract out to other agencies to be able to make this table. And so going through that process involves creating a request for quotes document, reaching out to multiple vendors before you can select one, and then going through the contract writing process. And while our contracting process might be unique to TNC, if other organizations are interested in restoration, they may consider a structure that is robust enough to withstand these environmental conditions. And so they also may have to contract out. And so understanding if they need to go through this process and how that impacts their timeline and their capacity. Laying out a field schedule. So in our timeline right now, it's a generalized field schedule so that we can understand how long is it actually going to take us to do this work. This here might be a little bit overwhelming to look at, but I just wanted to showcase that this is a snapshot of 1 week out of 10 weeks, essentially, that we have set aside for implementation between baseline monitoring, collecting, fragmentation, stabilization, stocking our nurseries, monitoring. So it really is a lot of work to lay out. It's important to look at this also to see where the community can fit in. It's important to keep them involved and intertwined as we go along. And so really looking at a 10-week process up front shows us how that they can participate, where their strengths are, and how we can bring them along in this journey. Outreach is also super important. This is an example of an outreach flyer I made for the work in Kealakekua Bay. And while we haven't started there, it's important to kind of reach out early, create those relationships with the commercial operators that frequent that area so that they're aware of what we're doing, why we're doing it, why it's important, but also how they can help and inform their guests to make sure that kind of everyone is following the avenues that they're supposed to do this work properly. It also allows us to be transparent with our community members in that each area. So if they're not directly involved in our planning process, it's still important that we let them know when we're going to be there, what we're doing, allow them to participate in our outreach efforts, and really just being transparent in all the work that we're doing. So what's next? We will continue our NEPA review. I have been scrolling through a 400-page programmatic permit and pulling out language that's relevant to our project so that we can be ready when we're able to get to the step. The work that we're doing is not only important for the ecosystems, but for our community as well. And so in each of our locations, we'll be doing an opening ceremony with the community members with hula and mele or song to really kick off the ecological and cultural importance of this work. And then fingers crossed, hopefully, as we're making progress on all of our permits, implementation in Ka'upalehu, end of this summer is really our goal. And so I just wanted to take a few minutes to, I'm super grateful for all the learning opportunities that I've had along the way. I've been able to attend the U.S. Coral Reef Task Force meeting, a Coral Reef Futures Conference in Florida, restoration workshops. I had the opportunity to go back to St. Croix in the U.S. Virgin Islands and help them with their coral outplanting. Earlier this year, I attended a training on TNC's kind of project management framework that I was able to use and apply to this restoration project. And I also helped Julia, my mentor, kind of plan and participate in a workshop for that post-storm response initiative with coral restoration. And with that, I would like to give a huge mahalo to everyone that has supported me, my friends and family, my mentor, Julia, my advisor, Lisa, all of the Hawaii Island Marine team, and my wonderful TCBES cohort members. I could not have done this without you guys. So thank you. Yeah. Questions? Do we have time for questions?
Yeah, I think one or two.
Okay. Yes.
Great
Yes.
presentation, amazing work. This is probably a really exciting question. I'm just curious. It makes sense your kind of rationale for using, of course, corals from the local area, and kind of corals of opportunity that are not already broken free. But I was just curious to hear your thoughts about the corals themselves that are chosen to be used in that places, that have very different kind of systems like Kaneohe or American Samoa, where you have these kind of active physics, and you have these like super corals that are really well adapted to these extreme temperature and extreme temperature fluctuations. Maybe it's not that informative, but I was just curious, like on a local scale, especially here, for these reefs on the Big Island, is there a difference, say, from a coral that you might choose that's in the very shallow, close to the intertidal, and maybe experiencing more temperature fluctuation versus one that's from deeper, further offshore? And you think that that matters at all, and it should be part of the planning, or is that a totally
Yeah. So just to repeat your question and make sure I understood it, like where we choose to collect corals from, that environment that they are growing in, how that affects they're able to withstand the different temperature flux, maybe make them more robust for what we're doing. Yeah. So I think definitely where we collect them from plays a big role in what they're adapted to. And so for this pilot project, we're kind of focusing on initial implementation of kind of collecting the corals that we do find, but it is an additional component that we are looking to work in, is figuring out how that plays a role in how successful they are in our restoration project. And so looking at the thermal tolerance of different coral colonies, either based on where they're from or looking at external factors. I'm actually traveling to Palmyra this summer to help a researcher with a thermal stress testing experiment, so that I can kind of learn the methods on how they do it and be able to bring it back to our restoration project and eventually implement that with future corals that we collect.
Yes. I saw in your outreach materials that you had a sign for your study out in the water. Have you thought about what kind of language you have out there so that people try and not mess with it or try and help, I guess?
Yeah, and so that was kind of one of our concerns, or like one of the factors behind outreach is making sure that people are going to be eager to help and want to do the right thing, but we just don't want that to look like them picking up random corals in the sand and be like, oh, I'm helping, and putting it on our table. And so thinking about signage for underwater is definitely something that we're considering, and figuring out kind of what the right language for that is to be more informative of like, hey, this is what we're doing, but also like, just so you know, without the proper like requirements, we can't have you taking corals and putting them on our table. But it's also important for finding other avenues and ways that people can help, so they're not resorted to wanting to pick up corals and putting them on our table.